All right, I've gotten the green light that we can kickstart the event officially. So, good morning once again, ladies and gentlemen. Ladies and gentlemen, good morning. Ladies and gentlemen, good morning. I'm bidding you all a warm welcome to this August gathering. By virtue of nomenclature, I remain Jimo Ridwanola Omeiza, a student from the Department of Microbiology. People call me Genie, a creature of magic. I call myself the guardian of the unseen galaxies, because literally that's what microbiology is. And it's of great delight and immense pleasure to welcome you all to this wonderful occasion. But ladies and gentlemen, I'll urge you all to fasten your seatbelts, because on this very event, I'm going to be like a virus, infecting every one of you here with unlimited entertainment, fun, and a whole lot of learning. Ladies and gentlemen, welcome to the maiden edition of the annual conference of National Association of Microbiology Students, NAMS ABU students, in collaboration with American Society for Microbiology. Can we please put our hands together? Ladies and gentlemen, I believe we can do better than that, so please put your hands together. You know, one peculiar thing with microbiology ABU is that even the structure of the department speaks volume. If you take a whole look, with respect to every other department in the faculty, the microbiology department looks more like a family house. It's a building where our lecturers take us as children, and we, the students, we take them as fathers, as mothers, and even as role models. So if you're proud of being a microbiology student, I want you to put your hands together again. Thank you very much. So quickly, on the order of events, we made a little bit of adjustment, so let me quickly clarify that. On the normal circumstances, we are supposed to start with the national anthem and the ABU anthem, but we interchanged it. So now we'll be proceeding to the introduction of speakers and the guests, after which we'll all rise up and pay our homage to our motherland as well as our citadel of learning. Are we together, please? Microbiologists, one house, are we together? Thank you very much. So, to the introduction of speakers and guests. Distinguished audience, it is with great delight and immense pleasure that I would like to introduce a special guest of honor in person of the Vice Chancellor, the leader, the individual who is channeling, the captain of our ship, who is channeling the ship of Ahmadu Bello University, Zaria, to the Graceland. And that is in person of Professor Kabir Allah. Although he is unavoidably absent, but the moment he comes or he sends a representative, we will honor the person and invite them to the high table. Moving on without delay, the chief host of this event is a mother to all. One of the very few individuals who motivated me with a few words upon entering to Ahmadu Bello University. You know, she said something that life will deal you a whole lot of vigorous blow. Life will get you disappointed at some point. But if life gives you lemon, instead of whining and crying, try and make lemonade. Distinguished audience, I would like you all to put your hands together as we introduce to the high table the chief host of the event, in person of the Dean, Faculty of Life Sciences, Professor Mariam Aminu. You're welcome, man. You're welcome, man. With our elegant strides like the lioness. We from Kogi, we know what lioness actually do. Thank you very much, man. You're welcome, man. Thank you very much. Thank you very much. Ladies and gentlemen, moving on to the host of the event, the head of one of the most prestigious departments we have in school. I'm not bragging now, but the facts are there. All right? I'm going to be introducing the host, a man of timber and caliber, like my friends will always say, a man of unparalleled excellence who has dignity that speaks volume. Distinguished audience, let us put our hands together for the host of this wonderful event, in person of the head of the Department of Microbiology, Professor M.B. Tijani. Keep clapping, put your hands together. When you see wisdom, when you see knowledge, when you see understanding, when you see a man who truly resonates the impact of microbiology just by his words, distinguished audience, I believe we should do more. Thank you. Thank you very much. Thank you very much. At the same time, one of our wonderful dignitaries here present is also worthy of noting, and that is the Deputy Dean of our humble faculty. Ladies and gentlemen, let us put our hands together for the Deputy Dean, Faculty of Life Sciences, in person of Professor Gadsama. Thank you very much. Thank you very much. You're welcome, sir. Welcome to the high table, sir. Keep clapping, keep clapping, keep clapping. Oh, all right. Thank you very much, ladies and gentlemen. You see, for somebody who was actually raised in the southwestern part of the country, I am humbled by the humility of ABU lecturers. When you see wisdom, knowledge, and understanding putting themselves down and living averagely without any further. We from the southwestern part, we know what I'm trying to say, so let's not delve further into that. Thank you very much for your attendance, sir. We really appreciate it. Also, ladies and gentlemen, another special guest we have here today is the Assistant Dean, Faculty of Life Sciences, and that is in person of Professor Abashi. Put your hands together, please. Put your hands together. Okay, sorry for that. He's unavoidably absent. When he joins us, we are going to continue. All right, now, moving on to one of the main guests we have in our midst today. You know, when I heard we are going to be out and all that, but ladies and gentlemen, the humility that comes with people we have in our midst today is actually worthy of applause. So, distinguished audience, permit me to invite to the high table the young ambassador to Nigeria of American Society for Microbiology, ASM, and the lead convener of this conference. We have a saying that looks can be deceiving. In a lecture hall, he can just walk in and sit amidst us, and nobody will know. But distinguished audience, in him lives a very big man. Put your hands for Mr. Ahmadu Bello. Thank you very much, sir. Thank you for coming. At the same time, we also have another member of ASM who is also in our midst today, and he goes by the name Mr. Habibullah Oladipo Jayola. Thank you very much. Thank you very much. Thank you very much. You're welcome, sir. Now, another important guest we have in our midst is the representative of the president of Ahmadu Bello University, Zaria Alumni Association. Ladies and gentlemen, he is also the deputy registrar of the Alumni Association. So put your hands together for Malam Bashir Munir. Thank you very much. You can do better than that. You can do better than that. Thank you very much, sir. Thank you very much, sir. Thank you for coming. Now, to the moderator of the event, my own mother. Forget the fact that we don't look alike. It's due to some genetic changes in the, it's microbiology with genetics. My own mother, the moderator of the event. Ladies and gentlemen, put your hands together for Dr. Habiba Atta from the Department of Microbiology, Ahmadu Bello University, Zaria. Thank you very much, ma'am. Thank you very much for coming. Thank you very much. Now, to the speakers, the wonderful individuals who will be sharing with us their wealth of knowledge. People who are ironed and tailored in the field of microbiology and also in the emerging trends in our society. Ladies and gentlemen, the very first speaker of the event is a professor of microbiology from the Department of Microbiology, Ahmadu Bello University, Zaria, in person of Professor S.A. Ado, who is going to anthem. Hello, ladies and gentlemen, let's have one house. Thank you. Would we stand? Cheering for our proud to set a sovereign motherland. Our flag shall be a symbol that truth and justice reign. In peace, oh, bow to honor and this
we can't escape. To hang on to our children, our banner without stay. Oh God, oh creation, grant these our one request. Help us to build a nation when no man is oppressed, and so with peace and plenty, Nigeria may be blessed.
Put your heads together for your motherland. Thank you very much. Let us please still remain standing. Let us still remain standing for the ABU anthem. Let us remain standing for the ABU anthem.
We proclaim from hills and valleys we come to draw from the stream of knowledge. Our duty is to succeed in learning and service. Excellence is our goal. Ahmadu Bello University, in our dreams we shall achieve. We give thanks to Almighty Allah, in Him we derive our strength. We beautifully shall have by the ways of good learning. Not to south, east to west, your name shall always be. Ahmadu Bello University, a citadel of great learning.
Put your hands together, ladies and gentlemen, and take a seat. Thank you very much. Thank you very much. So without further ado, let us proceed with the program. Now, the item we have on the list now is a welcome address by the chief hosts of the program, in person of the Dean, Faculty of Life Sciences, Professor Mariam Aminu. Put your hands together, ladies and gentlemen. Put your hands together. Thank you very much, ma'am. Thank you very much, ma'am.
In the name of the Almighty Allah, most beneficent, the merciful, the HOD of Microbiology, the Deputy Dean of Faculty of Life Sciences, the young ambassador of ASN, future microbiologists, you have to tighten your belt so that I will infect you with this welcome address. It is with immense pleasure and great pride that I welcome you all to the first annual microbiology conference hosted here at Ahmadu Bello University in collaboration with the American Society for Microbiology. This is the first of its kind. Today, we gather to explore and advance the frontiers of microbiological research and to push in the boundaries of what we know and applying our expertise in meaningful ways. Microbiology, as a dynamic and ever-evolving field, holds the key to numerous scientific and technological advancements that can transform industries, enhance public health, and address global challenges. If you know, remember when COVID-19 came, microbiologists were in the forefront. We are the ones that can do this. Our conference, this conference, the program is designed to foster in-depth discussion and stimulate innovative thinking across various aspects of microbiology. As you see in the program, the first topics we are going to discuss is microbial biotechnology and the roles of microbes in industrial application. See, microbiology is applied as you know, this particular topic, we highlight the extraordinary potential of microorganisms in driving industrial innovation and addressing practical challenges of this world. The second topic is computational tools in genomic surveillance and infectious disease epidemiology. And you know, we have an expert in the name of Professor Ebi Suleiman who is going to address you on that. He is going to explore how we can advance computational tools and reshaping our understanding of disease patterns and enhancing our ability to respond to infectious disease threats. The third aspect of this conference is preparing future microbiologists for emerging trends and technologies. This is very important. This discussion today of this topic, we focus on equipping the next generation of microbiology such as you with the skills and knowledge necessary to navigate and excel in a rapidly evolving scientific landscape. This is what you will learn today. In addition to all these three topics, we are excited. We are going to feature a debate by our able undergraduate students. The topic of the debate will be designing a sustainable future with synthetic biology. Will microbial engineering accelerate the STD progress or open a Pandora's box of unforeseen consequences? We are going to see this. The debate will challenge us to think critically about the implications of synthetic biology and its role in achieving sustainable development goals. I am sure the debate is going to be very exciting. So, as we embark on this intellectual journey, I encourage each and every one of you, you have to engage actively, listen attentively. You are going to share your insights during the question and answer time. You can build connections that will drive future collaborations and innovations. Let us embrace this opportunity. Impress that sometimes when we even have some conference, we don't see the hall as packed full like this. So I want to thank you for being here, for contributing to what promises to be a stimulating and productive conference. I look forward to it so that we will say one day that we had a conference in microbiology that will forever remain in our mind as a memorable one. Thank you all for coming. I welcome you to this conference.
Thank you very much, Ma. Microbiologists, I believe you can do better than that. We can do better than that. Life scientists, I believe we can do better than that. Put your hands together again. That was Professor Mariam Inu, the Honorable Dean, Faculty of Life Sciences, Ahmadu Bello University, Zaria. So, ladies and gentlemen, the next item on the list is supposed to be the brief remark by the special guest of honor, who is unavoidably absent. So we are going to be skipping that and moving to the next item. But please, I would like to use this medium to tell the performers from dance and drama troupe that they should please kindly get ready. Thank you. Now, the opening remark for this event is going to be given by the lead convener of this annual conference, the very first of its kind, in person of Mr. Yusuf A. Tajuddin, the Young Ambassador, American Society for Microbiology to Nigeria. Put your hands together, please. All right, before he commences the speech, please, ladies and gentlemen, put your hands together for Professor I.O. Abdullahi from the Department of Microbiology, Ahmadu Bello University, Zaria. Put your hands together, put your hands together. Thank you very much, sir. We really appreciate your presence. Thank you very much, sir. So, ladies and gentlemen, I present the Young Ambassador, American Society for Microbiology to Nigeria, in person of Mr. Tajudeen. Thank you very much.
The Honorable Vice Chancellor, Dean of Faculty of Life Sciences, distinguished professors on seat, esteemed lecturers on seat as well, and future microbiologists. It is my profound honor to stand before you today as the current American Society for Microbiology Young Ambassador to Nigeria. As a microbiologist and early career researcher, I have had the privilege of witnessing a remarkable progress and evolution in this great field of ours. But I have also seen many persistent challenges that many of you face while transitioning to this highly competitive world. My aspiring young microbiologists, I am realistic with you. The landscape of microbiology is evolving at a breathtaking pace. And while you are there on Twitter, posting after microbiology what next, this field has flown just like the world, leaving you behind. You see, the skills and knowledge that made us strive as students, the ability to essential, are not sufficient to excel as professionals in today's world. The truth is that the very nature of what we do in the field of microbiology is being rapidly transformed by advancement in technology, multidisciplinary collaboration, and overwhelming demand for microbiology to solve real-world problems. Let me share a personal story to illustrate my point. When I completed my Bachelor of Science degree in microbiology at the University of Ilorin, I had the opportunity to work alongside brilliant young scientists, a microbiologist to be precise. His name is Iola, the previous ASM Young Ambassador to Nigeria. By then, he had just completed his Bachelor of Science degree in microbiology and was seeking scholarship to pursue a postgraduate degree through the leading international vaccinology education Erasmus program. Now, Iola was no stranger to the laboratory. He had mastered the technical intricacies of antimicrobial sensitivity testing, genetic sequencing, and microscopic examination of laboratory samples. Despite the competitive nature of this program, what truly set Iola apart was his ability to think strategically, see the big picture, and effectively communicate his project findings to diverse selection committee members. He eventually got the scholarship, a fully funded one at that. And as I'm delivering this speech, Iola has completed his master's program, awaiting his convocation ceremony. So wait till then, we will send you the invitation letter. So Iola's story resonates deeply with me. I've also experienced the transformative power of leveraging one's expertise to make a real-world impact. Recently, I was awarded a research grant from the Royal Society for Tropical Medicine and Hygiene in collaboration with the National Institute of Health and Care Research in the United Kingdom to investigate the risk of spillover of zoonotic pathogens at humans-animals interface in a biodiversity hotspot in Ibarapa region of southwestern Nigeria. So the reason for this project is to prevent zoonotic pathogens of pandemic potential. So we don't want a rebate of COVID. The project wasn't just an academic exercise. My microbiological and public health knowledge were applied to understand the risk factor for zoonosis in this region. My team and I had to collaborate with the local communities, navigate complex cultural landscapes, challenges, and adapt the research methodology in our proposal to the reality on ground. Because when you get to the field, you realize that there are quite a lot of things that you would change, and then you need to go back to your methodology and do the needful. So the experience I had pushed me far on the confines of pathogen identification in the lab, demanding skills in project management, community engagement, and effective cross-cultural communication. So the findings from our work have been presented at the 2024 Planetary Health Annual Meeting in Malaysia. It was actually presented by Mr. Abibullah Oladipo. So the reality here is we did not pay a dime to attend the conference. We were funded. So these experiences, both Iola and my own, validate a critical point here. In the present day world, a microbiologist needs to be more than an investigator or researcher. We must be a problem solver, innovator, communicator, and becoming bridge builders between research and local communities we serve. My dear aspiring microbiologists, this is the kind of versatility needed in our field today. Yes, while it is expected of you to have technical competencies in bioinformatics, questions, or how many of you have struggled unraveling the complex multidisciplinary research where you find yourself speaking in a language different from your colleagues in microbiology, medicine, and public health? Realistically, the skills that made us thrive as students may no longer be enough for us in the outside world. And that's why it is highly imperative to seek out opportunities directed at bridging this skill gap. Internship, industrial training, and CWES, for example, provide unparalleled opportunity to gain practical experience and build a professional network. Mentorship programs are another invaluable resource. I've had the privilege of being mentored by professional microbiologists. Then, whom I cherish the most is my amiable Dr. Abiba Ilyasu Atta, who never for once ceased to offer that leg up. Thanks so much, Doctor. I was able to publish more than 30 journal articles in international peer-reviewed journals and won some research grants from international committees because she's always there to support. Beyond mentorship, engage in extracurricular activities and own your leadership skills. I'll never forget a time that I led a team of interdisciplinary researchers in addressing the risk of zoonosis in Ibarapa. Despite being a challenging experience, Ibarapa is actually a remote area. And I don't know if you've been there before, you know what I mean. So I learned the power of collaboration, the importance of avoiding silo thinking in research, and the value of community engagement to make a significant impact in our world. Aspiring microbiologists, the path ahead may seem challenging and daunting, but I so much believe that you can only see further by standing on the shoulder of giants. Who are these giants? These are lecturers, as they can only provide us with the right support that we need to bridge the skill gap and unlock our full potential in this field. Please forget the myths that people usually say that, oh, microbiology, they end up working as a POS attendant or whatever it is. It is how you prepare yourself that you meet opportunities in this field. So let us embrace the change in our field and rise to the challenge of becoming vibrant and impactful leaders that the field of microbiology desperately needs. If a great scientist like Liz Pasteur said, wavering commitment and dedication to making a difference. The future of microbiology is in our hand, and the time to act is now. Thanks, and let's get to work.
Thank you very much for that wonderful. I really don't know what to call it, because from what he said, I believe not only myself, but the majority of us here, I believe we are now seeing it's microbiologists in training, like we call ourselves. I believe we can see the path that lies right ahead of us. I believe I'm not the only one seeing that microbiology actually entails more than culturing in the lab. He said something that is actually worthy of note, that microbiologists are problem solvers. So in other words, the problems our world is currently facing, starting from the issues of climate change to antimicrobial resistance to different chemotherapeutic failures and all that, I think microbiology will really play a vital role. And let me ask you one more time, are you happy you are a microbiologist? Are you proud of studying microbiology? Put your hands together for yourself one more time. Thank you very much, ladies and gentlemen. During the course of the opening remark by the ASM Young Ambassador, we had some important dignitaries work amongst us. So ladies and gentlemen, permit me as I invite to the high table the third speaker for this annual conference, who also happens to be the head of the department microbiology, Kaduna State University, in person of Dr. Fatima M. Musa. Put your hands together, put your hands together. Thank you very much, ma. Thank you very much, ma. We really appreciate you. Thank you for coming. Thank you for coming. Also, ABU microbiologists, we have some of our wonderful lecturers work in. So I will call them and you guys will put your hands together while they wait for recognition. Ladies and gentlemen, currently I'm seeing one of my favorite lecturers. I don't have favorites actually, but she's one of the favorites that I don't have. Distinguished audience, put your hands together for Dr. Zainab Anchow. She walked in not long ago. Put your hands together. Put your hands together. Thank you very much, ma, for making it here. Thank you. You're so much appreciated, ma. Also, sitting right next to her, we have Mrs. M. Abdulrazak. Put your hands together, please. Put your hands together. Thank you very much. Thank you very much. And right behind them, I'm seeing one of our wonderful lecturers. He has a direct connection with we in 400 level. Distinguished audience, please put your hands together for Dr. Henry Bishop. Thank you very much. Thank you very much. You guys are doing well. You guys are doing well. Thank you very much. Thank you very much. All right, we also have some important dignitaries here as well. And permit me to introduce the President, National Association of Microbiology Students, Kaduna State University chapter, in person of Adamu Saminu. Can you please wait for recognition? Thank you very much, sir. Thank you for coming. Thank you for coming. Also, we have the NAM's Northern Region Governor, in person of Mr. Samuel Akaji. Can you please wait? Thank you very much. So let's move on with the program of the event, ladies and gentlemen. Permit me to now invite our father, is a father figure here at present. Ladies and gentlemen, let me invite the representative of the president of Ahmadu Bello University Alumni Association, who is also the deputy registrar of the Alumni Association, in person of Malam Bashir Munir, for a goodwill message. Thank you.
Good morning, ladies and gentlemen. The chief host, who is unavoidably absent. You have a lot of things to do as a microbiologist. Today, I was watching a newspaper, I was watching news in the television. I saw there is a new disease in town, the mpox. Was it mpox? Yes, I saw it. So these are issues that you are to embark on research on, and your discoveries are what the nation and the entire world would use in solving such problems. So we are very happy that you are doing your best. And on this note, I think it's not the time for speeches, but the national president asked me to pledge the sum of 50,000 to the conference, although he has already
Of microbiology. One of our fathers who really made us understand the basis of immunology. In person of Professor Ella. Thank you very much, sir. Thank you for coming. Thank you for coming. Also, we have one of our other a young and vibrant lecturer really doing well in terms of impacting knowledge into the younger generation. And ladies and gentlemen, allow me to introduce, while she waits for recognition, our honorable lecturer in person of Mrs. A.N. Suleiman. Thank you very much, ma'am. Thank you very much, ma'am. Thank you very much, ma'am. Also, one of our honorable fathers is here, a lab technician, one of the people that are actually training us and showing us the basis of microbiological practicals, in person of Madam Shitu. Put your hands together for him, please. Thank you very much, sir. Thank you very much, sir. Also, sitting beside him is worthy of recognition. He doesn't know me, but I know him a lot. He's a direct descendant of Professor Ahmadu Bello of biochemistry. And ladies and gentlemen, allow me to introduce, while he waits for recognition, Dr. Bash. Thank you very much, sir. Thank you very much for coming, sir. Thank you very much for coming, sir. All right. Now we are going to be having a performance by the dance and drama troupe. Are they ready? The dance and drama troupe, please. All right. Ladies and gentlemen, we are having a performance by the dance and drama troupe. I would like you all to sit back and enjoy them. Thank you very much.
Everything you know what you make and tell you go dey run. Why are you running? No, yes. No, they touch this man I give you. I don't call medicine man. They call. Wait, one thing. You don't tell me say I was for this thing. You don't tell me this thing. I will find you for yeah, the thing just well on the stomach of yam. We can don't worry, I don't know. For you, I know you because I will put us for his first. You go pay you, I don't pay you too much right now. Why you the one this now? Why this now? This is where you come. Why you go get your this? man dey do. Na him I take through my family till date here.
See, mommy, I've told you, stop finding small to you. What be meeting you? What does he know? What does he know? All the drugs that he has been giving me, why is he next?
Mo pẹ́, this leg wey you dey see like this, and this no wey you dey say. Even this oyinbo people wey you dey see say they know something. E get one thing wey they dey talk say, wetin be that thing? Say we na care we dey care, and na God dey heal. Na so me now I dey depend on God o.
He's a very good doctor. Yes, mommy, he has machine that you put it on your leg. Mommy, how that you put it on your leg? They will look at your leg. They will see what's wrong. See, mommy, they have machine that they should collect your blood. They can tell you have malaria, you have typhoid, you have anything that's wrong with you. See, mommy, there's even something in my biology chapter. She said they have one machine, call it microscope. Mommy, don't worry about it. Mommy, don't worry. We have machine. Anything that is wrong. See, mommy, she needs to see what I like this clean. That machine is there. If there is bacteria or microorganisms.
The machine has got.
This machine is not the best for anything else. We don't trust you.
Even you, because of you, I enter inside here. Don't see your wife. Because you don't see your wife. You don't know. I think to myself, I think myself over.
Put your hands together, put your hands together for them. Thank you very much, thank you very much. I believe
why did you have to wait until this time? Before bringing this step, madam. Looking at the way this step, I will have to conduct an accurate diagnosis. I think we should take it because this thing is looking like something that has been infected with some bacteria. Yes, I think I need to conduct a genomic surveillance. I am not wanting your leg. I just want to monitor and track how this leg has been affected and we need to just follow your husband carry
Thank God. As we do the thing, we carry the God to back us. Now, they give out. See, let me start up.
Now, sometimes they you see this man wey dey big camera, even the bacteria wey inside na why we need to be there. As na don come, this thing no go just go. This thing wey look like yam before, now you just say you don do.
Bacteria are very
Yes. We know say bacteria bad, but then make we also know say this bacteria wey we talk and microorganisms, we also call them for our own medical terms microbes. These microbes, we fit use them for our industrial age wey we dey now. We go fit use am to produce plenty things. Our goods wey dey valuable to us as humans, if we process all these microbes, all these things, we go fit use am do some things wey go make us get now. So when they ferment all these microbes, this bacteria wey you say that, if they ferment some of them, they go fit use am through that process, use am take produce food. Yogurt wey you dey do, umuaya wey you dey drink, if they ferment all these things, yogurt say that bacteria dey inside, but na because they turn am into something wey the vaccine wey we use treat your mama, and antibiotics wey she use, na the same thing na we see. So bacteria, if we use am well, when we come process am, produce am, reproduce am, we go use am do better. So you know dey bad. Hear me, I know say una no know this kind thing.
So don't go, you're saying that bacteria are bad, but when they go through the right process, they can become good.
Technological,
It's not
we need some strategies. You see, the first thing we do now, she wants come out from the system, and she won't enter higher education. Now, maybe for the school where we say they incorporate all these microbiology courses like genomics, microbiology, data science, analysis. I don't say you don't understand all these courses they get microbiology. When they we go carry them.
Which school you go?
Me and University of Ibadan but as one time, you get one anything, all this technology, if you go their lab, you may find out I am not all these function, mentorship self dey. Now when she finish, I go just rush enter school. As I dey now, I go fit carry her under my wings. I go dey teach her small thing wey she dey know, even before she enter. Because of that time function and seminar.
My people, what will buy for this? No.
Put your hands together, put your hands together. You know, when honorable lecturers and our mentors tell us that microbiology is versatile, it's everywhere. I can remember when I was in SBRS, Madam Aminu, they'll always tell you microorganisms are ubiquitous. They are everywhere, and their impact can never be swept under the carpet. You can see, these are students from the Department of Theatre and Performing Arts, yet they have ideas of vaccines. They know of microorganisms, they know of bacteria, they know of fungi. So ladies and gentlemen, I believe they still worth another round of applause, please. Put your hands together for them. Thank you very much. During the drama, some of our wonderful lecturers also came in. I would like to recognize the presence of Dr. Rosemary Ame. Thank you very much, ma. Thank you very much, ma. Thank you for coming. Thank you for coming. And also one of our other lecturer already on the high table in person of Mrs. Murja. Thank you very much, ma. Thank you very much, ma. Thank you for coming. All right, we are moving to the topic for the conference in proper now. So ladies and gentlemen, I said something earlier that the moderator of this event is my mother. So this is a mother and son event. So distinguished audience, allow me to introduce my mother in person of Dr. Mrs. Abba Atta to read the citations of the speakers. Thank you very much, ma'am. Thank you very much, ma'am.
Good morning, everyone. Our Honorable Dean, Head of Department of Microbiology, the representative of the President of the AB Alumni Association, invited guests, speakers, colleagues, ladies and gentlemen. I welcome you all to this first conference, hosted and jointly collaborated with the American Society for Microbiology. So, before I read the citation of the first speaker in the person of Professor A.B. Suleiman, I would like to encourage us all. Many of us do not know about the American Society for Microbiology. So we may only know about the parent body in Nigeria, which is NSM, Nigerian Society for Microbiology. But the American Society for Microbiology is free for any microbiologist to join, either as a student or as a graduate. So I encourage us all, even as students, please visit their website, www.asm.org, and register to join. And you have a lot of resources at your disposal to use. I remember the immediate past young ASM ambassador, the person of Mr. Iola. I met him through ASM. We have never met face to face. Same thing with the current ASM young ambassador, Mr. Yusuf Tajuddin. We met during COVID. So when the MC said in the beginning that the Dean said, if life gives you lemons, make lemonade, my example of meeting them is a very perfect example, because we met during the COVID-19 pandemic. And how did we meet? We met online through ASM. And today we have forged a very strong partnership and mentorship relationship. So I will urge you guys to please register on ASM and have a lot of resources, networking opportunities, and so many things at your disposal. So our first speaker today is Professor A.B. Suleiman. Professor A.B. Suleiman obtained his Doctor of Philosophy degree in Veterinary Public Health and Preventive Medicine in 2011 from the Faculty of Veterinary Medicine, Ahmadu Bello University, Zaria. His bachelor's degree was in microbiology from the Faculty of Science, Bayero University, Kano, in 1995, while his master's degree in medical microbiology was from the Faculty of Medicine, University of Jos, in 2003. So you can see that he's a very multidisciplinary scientist. Professor Suleiman started his career of over 25 years of teaching and research at the prestigious National Veterinary Research Institute, VOM, Plateau State, Nigeria, in July 1999 as a research officer too, and later moved to Ahmadu Bello University, Zaria, Nigeria in 2013 as a senior lecturer. At the university, he taught many undergraduate and postgraduate courses. He He has supervised students and researched extensively, particularly on microbial genetics and epidemiology of infectious diseases. Jokingly, some of us like to call him Mr. Molecular. He has over 70 journal paper publications and has attended many conference proceedings. Professor Ebi Suleiman has attended over 15 major national and international scientific and professional conferences. He's a member of the editorial committee and board of 2 learned journals. And in the process of his career, Professor Suleiman has supervised and graduated many masters and 15 doctoral students. I think he deserves a round of applause for that. Professor Ebi Suleiman has served as an examiner at the undergraduate and postgraduate levels in many institutions. For instance, Bayero University, Kano, Abraka Tafawa Balewa University, Bauchi, Umaru Musa Yar'Adua Katsina, Nigerian Defense Academy, Kaduna, and Kaduna State University. He has also served in many committees as a member or chairman of curricular development at Kaduna State University and also at Federal University, South Zamfara State. And he has been involved in a lot of activities. So the major one at the national level, Professor Suleiman has served as World Health Organization Northwest Zonal Consultant on poliomyelitis eradication. He served on this committee between the years of 2015 and 2019. He has also been a member of COVID-19 technical team at Ahmadu Bello University, Zaria, 2020. He has been a member of the National Committee for the Verification of Measles Elimination from 2017 to date. He is also a member of the Nigerian National Mirror Committee, also the Nigerian National Technical Committee on Biotechnology from 2023 to date. So his major research interests and area of specialization are microbial genetics, molecular epidemiology, and bioinformatics. Professor Suleiman is happily married with children. Thank you. Please join me in welcoming him to the stage.
What is it? Huh?
Dean, Faculty of Life Sciences, Head of Department, Microbiology Department, other very important people here, lecturers and researchers, welcome to this presentation. Sorry, I forgot the convener himself, the ASM ambassador. Sorry for that. So I'm going to make my presentation, which seems very little. But actually, my presentation is to prime you. My presentation is, as it was said by the Dean, to bring an insight and to provoke stimulation in new area of research. Simply to make you think, simply to make you look at things from another angle. In fact, this is a new era, this is the future for you. So the title of my presentation is Computational Tools in Genomic Surveillance and Infectious Disease Epidemiology. Really, I wanted to change the title, then I remember, no, I'm not the one doing it. Let me just do it the way it is. And actually is knowledgeable. So let's understand first, what is genomic surveillance? I want us to look at it from perspective. Focus is monitoring the genetic diversity of pathogen in real time. For example, it must be continuous in that perspective, meaning continuous and then in contiguous manner. Then what are the data? What do you use if you are doing genomic surveillance? What you need is DNA, RNA sequences, because that's where the information will come from. That is where we have everything about that particular organism. If you remember your DNA replication and stuff like that, this is what we are going to get. So the other one is timing. No, sorry. The other one is how do you use genomic surveillance? Are you going to use genomic surveillance for prevalence studies? No. You have to use it for incidents. Now, and that may be one of the demerits for this particular thing. But the thing is that whatever you're getting from it is real. There is no shushing, no ambiguity in that. Let's go with the slide. Now, what is infectious disease epidemiology? Studying the spread and control of infectious disease in population. That is at population level. Here we're talking about classical epidemiology. That is what we're talking about. So that is the goal. And then that is the focus. The understanding is understanding epidemiology of diseases, identifying risk factors, and evaluating interventions. You know, the understanding, the study of diseases and the dynamics within a population where you get susceptible, resistant hosts, the dynamics of transmission itself is simply the epidemiology. And that is the goal for infectious disease epidemiology. Now, what are the design? Design simply, we're using things like cohort studies or case studies. These are basic epidemiology. That's the design. If you are talking about data collection, what kind of data? We have three kinds of data. One, epidemiological data. That is when you get your case report, you can use questionnaire, you are getting epidemiological data. Then you have clinical data. Clinical data, the clinician will clerk the patient, will actually say, okay, this is what is happening, and you have a record. Then we have the laboratory data. Somebody goes to the laboratory, collect blood, whatever kind of specimen, and he check what is there, and that is recorded. These are the 3 points, what we use to actually know if that thing is there or it's not there. Now, the other aspect is technological data. An example is all this case report, contact tracing, environmental data. If you remember during SARS-CoV-2, that was a very important thing they did because it's a new organism. Nobody knows its source. Nobody knows where is it actually coming from. And then we don't even know the intensity or the transmission dynamics. So we use contact tracing. Have you met somebody who got the disease? How many people have you met? You, if you go back home, how many people are you going to meet at home? So that was what they used. And once they realized you met somebody with the disease, what they did then was to isolate you. The idea is to cut transmission. Now, for this epidemiological data, you can use historical prevalence. You can use current, and you can forecast. That is one of the advantages over the genomic surveillance. Next slide, please. Now, computational tools, what are they? Now we've gotten the picture of what are genomic surveillance and epidemiological infectious disease epidemiology. So these are the major two now. Computational tools. Go ahead. What are they? We say they are software programs or algorithms that use computational methods to analyze, simulate, and visualize data. What we need to understand here is there are different types of algorithms. And normally this algorithm is written. Most algorithms, they have steps you follow. You impute. After imputing, it's processed. After processing, the output will come out of it. Then later we'll look at the fitness of it. Then we'll look at the reproducibility. These are part of algorithm. You need to come up with this kind of thing. I will have a lot of many examples of algorithm at the gene bank and whatnot. We have a lot of them. Now, what we must understand, and is very important, is simulation, modeling. In this case, we're trying to take real-life example, real-life situation, come to the lab, use our computer, and put whatever parameters we need. Is it a population? Is it of temperature? Is it intensity of the infective agent itself? We model it so that we can be able to do what? Predict. Once we can predict, then we can be able to cut transmission later in life. So what are they used for? Solve problem. Yes, that's what I like with this. Computational tools are meant to solve problem. They are meant to give you an insight on how to do it. If, for example, I ask the first people that are seated here on the first row, go out and come back, sit, go back to your seat. Before they go back to see, if I ask somebody here on the high table, who sat there, who is number 1, number 2, number 3, you forgot. Now we are talking of DNA or RNA sequences of about 1,000 KB, and you are comparing them with hits that in the database, more than 1,000 or 2,000, and each of them will be aligned with each of the 1,000 or 2. You come up with the homology, you come up with the similarity, and then you give inference. Man is not so special to keep such kind of data. It's only machine that can keep such type of data, and that is why you're using this kind of tools. The other thing is prediction. Yes, if you can simulate, we can understand what's going to happen tomorrow because of so and so, based on what we have done, then we can be able to predict. Yes, we have an organism, it's affecting social number of people. This is how long it's going to survive. This and these are the susceptible hosts. This kind of environment will get it to grow by so forth. Then we can be able to predict the future, and we can use this information between officials and researchers to cover up these menace. The other one is get insight into various fields. You believe me that today, in Aki, people who are into building or designs or other things, they use softwares. It makes it easier for you. It gives you an insight. Things you don't have to do, things that you can spend 1 month to do, today you're going to spend minutes or seconds to get it. So software is one important aspect in our life. And believe me, more is coming into play. Next slide, please. So let's look at computational tools that are used in genomic surveillance. What you must understand, if you want to understand pathogen genomics and diversity of pathogens, there are 2 points you cannot run away from. One is assembly and annotation. Assembly is simply reconstructing. Reconstructing, you can only get it if you are using certain softwares. That's phylogenetic analysis. How is it related? Where is the source? How does it come into being? Some of the commonest software we use are SPADEX, that is for assembly. SPADEX is in Petersburg assembler. Proga is for annotation. You know, annotation is not about reconstructing. Annotation is about giving functions to different aspects of the genome. Where are the genes? Where is the regulatory gene? Where are the mutations? You need to use word annotation. How does it work? For example, with SPADEX, it's a software. It uses the same different kind of algorithm. Because one software can have many algorithms. We'll come to some later. SPADEX, for example, what you need, you just need a fragment of a DNA. For example, you just have a DNA, a fragment. That's just a read. And then you impute. Now this read will be constructed using the software so that we can have a complete sequence. Now this sequence can now represent the whole of a genome, meaning the only information, that is the output now is the sequence, meaning the only information you require for a complete genome that is missing, you can actually get it from that reconstructed sequence, from just a read, a fragment of DNA. You can get all the information required from that. Then the other one is Proca. Proca is just a prokaryotic annotation software. They call it Proca. Here, what we are doing is to get a genome that is not annotated. Simply, we don't know the function. For example, when you use databases, like something database, what you have there, we have annotated, deposits of sequences. Primary database, we don't know anything about it. So if you want to come up with annotation, you have to get something from a database, primary database, and then you put it into the program, that is the input. What you are going to get as an output is annotated genome. Meaning, if you take the genome, you'll be able to know everything about it from the points where the reading frames are, meaning the genes are, and then the mutations that are found on it, so that you can copy another one. So you have a complete picture of whatever you want to do, how to control it, how to use it for different kind of things. That's only on annotation. Then you have genomic epidemiology platform, like Nestrins. You know, Nestrin actually is a software. It's a software for viruses, different kind of viruses within an environment. This software has the potential to use algorithm and some mathematical models to compare the different phylogenetic traits, because it means to construct. Constructing is building something out of something. Reconstructing is there, you continue, you build something out of it. and then it compares the whole viruses that are there within that platform and gives a picture, visualization of how different they are and how they are relating. For example, they are thinking of a niche and environment. Now, the other one is computational tools for infectious diseases. This is basic knowledge. And all of us are using this thing. But we have a lot of those very strong softwares, like R. R, just the capital R, is a software for modeling. And then we have the Python library or Python programming. It's actually a software with algorithm with statistical and mathematical models. And it has something that is so important, knowledge decision, meaning it can learn from the data and it can give out an input that has not been programmed. That is what we call Python. People use a lot of Python for a lot of work. It's a software. Machine learning, we have the CK. I think I will discuss machine learning later. Then we have data visualization. Pandogo. Pandogo is a software for a very important for machine learning, and actually data visualization. Data is simply a kind of graphic representation. If you are using manual thing, maybe a distribution of the ladies in this place and the men in this place and different age, you can bring them into bars, pie chart, and whatever, so that you can bring the picture at a glance, you can know the difference. We have more men here than more women. Now we do the same thing using genomics. We get the picture of our virus, we take the whole genome, we sequence the genome, and then we don't have to even compare it using the database. We can now put them together to align. By aligning, we see the whole picture, we see all the areas of mutations. We see all areas for similarity. We see areas that are not mutated, and then we see areas that have repetitive sequences. Now, all these things, they have their own importance, both in clinical and epidemiology. If you look at it at a glance, you can give a picture directly. So these are very important things we use. Please, on this slide. Now, let me give you an example of blood example we had some times back, that is SARS-CoV-2. If you remember, during the SARS-CoV-2, there were a lot of theories. There were a lot of stories about it. When you use salt, you drink a lot of salt, when they started, you're going to be okay. You are getting this thing from different kind of animals, some from snails, some from where. I remember one American president who said the Chinese virus. You remember? Well, what they did, they make a total studies of this particular organism using software. This software they use gave us the whole picture. We're able to know the genome site. We're able to know who and who can get it. We're able to know the point, the receptor site in the host, why it attacked. All these pictures are done based on this program. Genome analysis, genetic analysis, and whatever, onto data sharing. That's the final point. What I want to discuss here is on machine learning. Machine learning actually is a subfield of artificial intelligence. It uses algorithms and statistical modeling. It enables the machine itself to learn from the data. You give it a data. The machine is not explicitly programmed. Now the machine will learn from the data. And now the machine will be able to predict or take decision on the data. Can you see that? At the same time, over time, the machine will perfect on how to play around with that particular data. Don't forget, the machine learns from the data. And then it does a lot of things on the data. That is knowledge discovery. Knowledge discovery is part and parcel of the AI machine itself. So you have to understand it. Now, I can decide to design an algorithm for my own use. I've been working with Star Warriors. I've been working with veterinary important organisms. I can decide to design an algorithm that can compare both the human and the veterinary bacteria that are associated with the guts, producing the guts. Now, my machine can be so useful to me, you can actually use it. If you're using the OSS software, you can modify it, you can use it, you can do whatever. But the way I will define my own machine may be different from the way you want to define your own machine. Now, this is the point. You have to understand this so much. In the area of bioinformatics, if you remember, the objective of bioinformatics is to design software. There are guys who are doing that. We have computer scientists. We have a lot of people doing that. For example, somebody is not here. I attended one conference with him somewhere in South Africa. Me, one of our lecturers here. The person who taught me genetic analysis, I want to be a musician. So it's an interest. The issue of bother and microbiology, biochemistry, mathematics, what and what now? Now we come together to solve the problem for ourselves. So we have to learn other things more than just what we know. We need to expand our horizon of knowledge. So that is machine learning. It gives you an insight into whatever you are doing. Because the machine will learn from you. And later the machine will use you or give you information about yourself and what you're supposed to do. We cannot run away from it. That is one of the important computational tools. Next slide, please. Now, why GS and ID? GS here, I mean genetic surveillance, and ID here, I'm talking about infectious disease, epidemiology. Enhanced resolution. I mean, there is no argument if you are using GS to actually identify your organism. There is no, because you are looking at the diversity. Because you are looking at points where it's not duplicated anywhere. You are looking at what is made of that particular organism. So there is no argument. The IDE will give you transmission dynamics. If, for example, you are infected with SARS or Ebola, how many people will you infect as a secondary infected person? If we're going to control you who is infected, then the number of people that will get the disease will be reduced. So understand transmission dynamics here is very important. Meaning we can do our secondary infected persons, we can reduce the number. How can we get that? We can only get that through IDE. And then even the position you are sitting in the room, how do you allow yourself, or how will you get the infectious agent? We have to get the design. In fact, in some studies with SARS-CoV-2, if you look at literatures, they even draw the housing. How the housing is supposed to be. How many people are supposed to be in the room? How many are supposed to be in the chamber? Now, ask the question. In our own setting, where you have 5 or 6 families, they are staying in the flat. How do you control the infection if you are talking of contact tracing? Now, these are very important points you do, and these are very important points you look if you are using IDE and IgE. Antimicrobial resistance, today we have softwares, very important softwares that can detect mutations based on antibiotic. Meaning, if social organism has social type of antibiotic resistance, there is a possibility that in future, this organism can become multi-drug resistant or can become another bug. There are softwares that can study this. Why that? Is because these antibiotic resistance are mostly mediated by plasmid or transposons. So if we look at the ability of the organism to absorb, look at the organism, the ability to absorb, stick on other extrachromosomal DNA, then we are talking of what? In the next 5 or 6 years, in a certain condition, after we model, we'll be able to know how this organism will change to become multi-drug resistant or even become a different fragment. Now, this is the idea of IDE MA or IDE AI. Sorry, maybe what I'm saying maybe sound funny or some kind of Greek. When you look at it very well, the idea is I want you to explore other areas so that you can check. Inform public health policy. If we have a good understanding between the researchers and the public health officials, and we have a good understanding of prediction, then we are going to save the whole world from a lot of menace in terms of health studies, in terms of public health, epidemiology. Please, next slide. Now, computational tools that are used for integration. Yes, you have 2 points. You have GS, you have IDE. How do you bring them together to work? What percentage of this will you bring in, and the other ones, so can we have optimum in the usage? And that's why we have other things like integration, we have gene banks, and then we have things like BEAST. BEAST here is taking variation estimation of species trees. We use them for different kind of work, and it's simply to estimate and to do what polygenetic analysis so that we can construct, so that we know what's going to happen. Sorry, I don't have much time. Next slide, please. Now, let me tell you something. I'm just introducing into the future. The trend is about the future now. The trend up is about a different leap about whatever you are studying. The world is now doing no longer in microbiology about chemistry or the mathematics you used to know. Now, what they are saying is machine is more important. It can teach you a lot of things. You can do a lot of things with it. So in conclusion, by leveraging computational tools, researchers and public health professionals can efficiently analyze and interpret large-scale genomic data, ultimately strengthening infectious disease surveillance, outbreak response, and global health security. Thank you for bringing me here.
I believe we can do better, ladies and gentlemen. I believe we can do better. Put your hands together. Okay, you know, I said something earlier that microbiology is everywhere, okay? And I'm saying SPADEX, I'm saying MicroReact. These are softwares. So basically, as a microbiologist, I can easily go anywhere and tell people that, see, guy, my work is not only limited into the lab. I can decide to sit on the system, do some genomic analysis like our wonderful resource person, our prof, said just now. And from there, right in the comfort of my home, I can solve a whole lot of problems. Isn't that wonderful? I can't hear you. Isn't that wonderful? Put your hands together for Professor A.B. Sulaiman once again. All right, during the course of our professor's presentation, the representative of the special guest of honor walked in. And distinguished ladies and gentlemen, it is of great delight and immense pleasure that I invite to the high table the representative of the Vice Chancellor, Ahmadu Bello University, Zaria, in person of the Deputy Vice Chancellor Academics, Professor Raymond Aku. Thank you very much for coming. Thank you very much, sir. Thank you very much, sir. You're welcome. We can do better. This is the representative of the Vice Chancellor. We can do better. Thank you very much, sir. Thank you very much, sir. You're welcome. This is the League of the Titans. Standing here, I'm already intimidated because the knowledge emanating from my left side, my friends will say the knowledge emanating from the 9 o'clock is knowledge. Thank you very much once again, sir, for coming. You're so much appreciated. Also, we have one of our lecturers walked in person of Dr. M.A. Shaibu. Put your hands together for her. She's on the item already. Put your hands together. Thank you very much. She's actually one of those who know me by my tribal name. So she's also my mother. Thank you very much, ma'am. Thank you very much, ma'am. So now, moving on to the next topic, I'll be inviting my mother also to come read the citation of the next speaker. Thank you very much. I'll put your hands together for her. While we sort out a little bit of things here, please, if you have questions, because I believe we should have a whole lot of them from the first presentation, how we can use some of the tools and all that. So if you have questions, kindly pen them down and pass it to any of the ushers walking around. Thank you.
Please permit me, before I call on the next speaker, I would like to invite the representative of the Vice Chancellor, the DVC, Professor Raymond Baku, to give us a few comments, goodwill messages to the students, because he has another function to attend. You are welcome, sir.
The Dean of the Faculty of Life Sciences, President of the Students' Union, who are the organizers of this collaboration, former Dean and HOD Microbiology, my fellow Fulbright scholar, Professor Miriam Aminu, professors, heads of departments, our great students, good morning. I bring you greetings. I was in my office working not quite 10 minutes ago when I saw his message to me. And he simply said, go and represent me at the ongoing conference in the Assembly Hall. So he forwarded the flyer, and I looked at it. I checked for the time. There was no time on the flyer. And I was wondering, I looked at my time, so it was after 11. And I said, okay. So I sent back a message to him to say, I'll find out and see what happens. So quickly, I called Professor Miriam Aminu, and she confirmed that she was here. And I said, well, you people have started, you have gone far. I don't want to come and disrupt the proceedings. Allow me to continue my work while you continue your business. But she insisted that I needed to be here. And that is why I'm here. Let me first apologize for crashing in while the program was already on. Not in our character to skip any important program like this. The Vice Chancellor is invited because the university take this intellectual exercise very seriously, and we encourage this sharing of information and sharing of knowledge. We want to note that the theme for this conference, which is put together by the Nigerian Association of Microbiology Students in collaboration with the American Society for Microbiology, has a theme advancing knowledge and leveraging relevant skill sets. It is very important that as you listen and as you learn from the resource persons that have been put together, that the world today is about the acquisition of skills and how you can use those skills to change society and the human experience. So as microbiology students, I urge you to continue to make yourselves amenable to learn new skills and to develop appropriate methodologies to research challenging issues relating to health and to apply that to solving real-time situations. Like you have been told, I have other commitments. I will excuse myself. I want to wish you very fruitful deliberation, and the faculty and the student body should be rest assured that the university at all times will support efforts like this for the advancement of knowledge. Thank you and have a wonderful conference.
Life scientists, I believe we can do better. Put your hands together for Professor Raymond. Yes, he has another commitment. So before he leaves, members of the high table will take a picture, a photo with Professor Raymond Baku, the Deputy Vice Chancellor of Academics.
Thank you very much. Thank you very much. The
Deputy Vice Chancellor Academics in person of Professor Raymond out of his busy schedule, he still took time to come say hi to us and give us a message of goodwill. So I think he deserves another round of applause, please. A better one at that. We heard he got the message about 30 minutes or less ago. So now we are going to continue with the paper. I believe we've learned something related to microbiology and how we can progress as future microbiologists. So the moderator of the event, in person of Habiba Atta, she's required here of the second speaker. Thank you very much, and put your hands together for her.
The second speaker we have is Professor S.A. Ado, who is ably an industrial several courses at undergraduate and postgraduate levels. He has also supervised over 50 master's students and 30 doctorate theses. Round of applause, please. He has attended several national and international conferences and has over 70 paper publications in national and international journals. Professor Ado has served in various capacities within and outside the university, including the head of Department of Microbiology, the Deputy Dean of Faculty of Science, the Acting Dean of Faculty of Science, the Coordinator of the University's SciWest program, member of the Science Complex Appointment and Promotion Committee, member of the University Library Committee, author project sub, member of governing board, Ahmadu Bello University Teaching Hospital, member of governing board, Veterinary Teaching Hospital. He has been a visiting professor and an external examiner to several universities. Professionally, Professor Ado is a current vice president of the Nigerian Society for Microbiology. He has also served as the editor-in-chief of Nigerian Journal of Microbiology. So he's happily married with children. So even though he's not here today, Dr. Bishir is also another mentee of Professor Ado in the same field of industrial microbiology. And I believe he will do justice to the topic. So join me in welcoming Dr. Bishir to the stage.
In Faculty of Sciences, Head of Department, Microbiology, the young ambassador ASM, all professors present, my respected lecturers, fellow colleagues, students, members of the press, security personnel here present, ladies and gentlemen, good morning. Before I proceed, I will have to ask you to forgive my voice. I'm in some cold for 2 days. I will be standing in for Professor S.A. Ado, and I will be speaking on the topic microbial biotechnology and the roles of microbes in industrial applications. These are the outlines of the presentation. We're looking at what microbial biotechnology is all about. And we'll be looking at the roles of microorganisms in industries. Also, we'll be looking at some of the challenges and limitations of microbial biotechnology. Microbial biotechnology is simply a field that encompasses the application of microorganisms in solving problems and creating innovative solutions across various fields. These field leverages on the unique metabolic potentials of bacteria, fungi, and other biofuels, pharmaceuticals, cleaning up environmental pollutants, and thereby creating a lot of possibilities in wealth creation and advancement in human well-being towards solving global challenges. Some of the roles or applications of industrial microorganisms raw materials into desirable products. Good example is that yogurt is usually made from milk, either powdered or fresh milk, and bacteria such as lactobacillus ferment the milk to produce yogurt that is having some unique characteristics, as a probiotic for people that are lactose intolerant. Also, the bread we consume is, as I said, a product of fermentation, yeast, which you call Saccharomyces cerevisiae. It does the fermentation and thereby producing some gases and some products that impart texture and an aroma in the bread. So the gas produced is the one that results in leavening the rising of the dough of the bread. And at the same time, when it is consumed, it increases health because the yeast is also rich in proteins and other vitamins. Now to the field of biopharmaceuticals, microbial biotechnology is also considered a cornerstone of biopharmaceutical industries, where microorganisms can be genetically modified, produce therapeutic proteins such as insulin, growth hormones that are used to treat various diseases. Before now, diabetic patients, which recently I came to know that about 4% of Nigerians are diabetic. So out of every 100 people, 4 of them are diabetic. None of the drawbacks is that the treatment is expensive. But today, because of the advent of microbial biotechnology, microorganisms such as Escherichia coli, which we commonly call E. coli, is usually engineered with the gene responsible for production of insulin. And therefore, within very short period of time, you can get thousands or millions of liters of insulin which can be administered to diabetic patients. And this approach, we said, provides a reliable and scalable source of essential drugs. Because without this genetic engineering of microbes to produce these therapeutic drugs such as insulin, before several cattle, for example, had to be sacrificed in order to extract the insulin from the islets of Langerhans in the pancreas. So, but today, you don't need to do that even in the laboratory, even in your test tube. The E. coli is engineered with the gene for synthesis of this protein, then it can produce large amount of insulin for application in diabetic patients. Now to the field of agriculture, microbial biotechnology has also found application. I believe most of us are already aware that we have bioinoculants, which usually are used as biofertilizers. Good example, Rhizobacterium species, which is used in order to enhance nitrogen fixation, nutrient cycle, and plant growth, especially in legumes such as peanut and cowpea. And I have to mention at this point that presently one of my students who happened to be the MC of this program is also working on this project on how to enhance nitrogen fixation by Rhizobium in legumes. And also, microorganisms not only are used to enhance nitrogen fixation, which I also need to mention mentioned that instead of buying these expensive inorganic fertilizers which are chemically synthesized and they end up being washed down into water bodies, then there may be eutrophication because of increase in nitrogen content, thereby resulting to algal bloom, which reduces oxygen concentration, killing aquatic lives, and then fishermen are having economic loss. Then we use these biofertilizers in order to improve plant growth. And biopesticides are also made from microorganisms instead of using chemically synthesized pesticides, we use microorganisms. One of the commonly used bacterium is Bacillus thuringiensis, which is usually used to system. Now, other applications or roles of microorganisms in industries include production of biofuels, and we said these microbes are key to the production of biofuels, such as they offer sustainable alternatives to fossil fuels. And those include the microbial fuel cell, and this is a sketch diagram of a microbial. During chlorination, the chlorine will not also be bio-based. You can prepare them from agricultural residues. These bacteria feed on the voltmeter, now generating at the same time because they are reducing organic matter content in the wastewater. So before now, the organic matter is going as a waste because the bacteria feed on it, now releasing some other undesirable byproducts. But today, bacteria can feed on the organic matter, producing electrons that can be tapped as electric current, which can be used to power some devices. Now, this is more like a flow chart of how the microbial fuel cell takes place. And this, at the anode chamber here, microbial oxidation of the organic matter takes place. And this orange bacteria, usually there are some that are more electrically, I can say, electroactive. Example is Schwannella onedensis and Eubacter species. They usually feed on organic matter and generate electrons. But other bacteria may equally do the same, especially Pseudomonas, but the issue is that the system needs to be completely anaerobic, and Pseudomonas is an obligate aerob, so it cannot lie here. Now, when the electrons are generated, then they are delivered to this electrode because the organism has no alternative source of electron but the electrode material. Some researchers use graphite, but because of low surface area, we therefore have to produce our own electrode materials with higher surface area. Usually here at the cathode chamber, it has been aerated, but in this case you can leave it as open air cathode. No need for aeration because during pumping of oxygen, so this can be an open air cathode which has been constantly aerated. This is a cathode electrode, and the proton generated is usually passing through this permeable membrane into the cathode chamber, which then the oxygen electricity instead of the amount of consuming in activated sludge system. It is believed that even in the United States, about 2% of their annual budget, which runs into billions of dollars, is going only for aeration of activated sludge systems. Therefore, microbial fuel cell technology is a recommended way of treating wastewater and at the same time tapping some chemical energy into electrical energy. Now, in the field of bioremediation and environmental application, I have already talked about wastewater treatment in the previous slide. And we also have what you call bioaugmentation. Naturally, there may be some organisms in the soil that can get rid of toxic pollutants and spilled oils, but we tend to get specific organisms that are very active, and then we put them in the soil or water in order to enhance, bio-augment the activity that is naturally going on. Also in the field of bioremediation of spilled oil, which I think is also important, that certain microorganisms are able to degrade some hydrocarbons, minimizing environmental damages. Now, in the field of enzyme production, microorganisms have also found enormous application, ranging from production of enzymes such as amylases, proteases, and lipases also, which have found application in dairy industries, even detergent industry, because in the detergent that we use usually contains enzymes. That's why if there is oil spilled on your clothes, when you put a detergent, then it will get rid of it because it might have contained lipase. Yes, so even in detergent industries, enzymes of microbial origin can be applied. Also in the synthesis of bio-based chemicals, ranging from bioplastics, biofuels, biopesticides, and biopharmaceuticals. You know, plastics, the synthetic plastics usually takes hundreds of years to degrade in soil. Therefore, they have become a menace in the environment, and that's why there is need for us to come up with a way of producing plastics, microbial biosensors, and diagnostics. So in the diagnosis of certain diseases such as diabetes and cancer, microbial biosensors can also be used at laboratory to scale up the system.
Ladies and gentlemen, we have our students here, and they are going to be presenting. They are going to be arguing that yes, synthetic biology will accelerate the goals of sustainable. Will accelerate while it's going to give a distinguished audience. Permit me to introduce the first speaker, who happens to be the first protagonist of synthetic biology, for him to come over here and present his argument in favor of the motion. Thank you very much. You have 5 minutes, please. 5 minutes. Thank you.
Vice Chancellor, Dean, Faculty of Life Sciences, Department of Microbiology. Ladies and gentlemen, my fellow students, I'm Emmanuel Kojo. Today I'm going to use highly intellectual and innovative arguments on highly existing biological systems, or edit or create new ones, solve or create specific functions and products. Synthetic biology, particularly microbiology, with countries, building capacities to implement these goals. Why then is synthetic biology or microbial engineering a better option than this other option? It's because microbial engineering has the potential to give innovative, efficient, precise, and minimize risk. This effectively solves the problem of clean and renewable energy. Also, synthetic biology offers innovative solutions, carbon capture and utilization, enabling the conversion of CO2, value chemicals, and fuels. This technology has the potential to reduce greenhouse gas emissions and help us meet our climate targets. Furthermore, in the case of hunger, hunger is a prevalent problem that humans have faced from time immemorial. Synthetic biology can improve crop yields, disease resistance, and these achievements through various applications, which include bioremediation, biosensors, biofertilizer, bioenergy, and biomanufacturing. By harnessing the power of GMMs, we can create a sustainable future for that energy. I trust that my arguments have been clear and convincing. Made a strong case for the importance of synthetic biology in addressing this. As we ponder on the possibilities of synthetic biology, let's ask ourselves, can we afford to ignore its potential?
I'm a 300 level student of microbiology department, and I'm here to oppose the motion that says designing a sustainable to microbial engineering accelerate SDG progress. Technological advancement is well welcomed, especially towards synthetic biology, particularly microbial engineering, as it holds a promising effect to agriculture, medicine, and etc. But the person I'm opposing, I beg your pardon, gave his view that this microbial engineering may pose a significant will improve resources efficiently, but I'm here to oppose that motion because we have to significantly look at the risks of these microbes that are being engineered. And so it's going to open a Pandora box. And a Pandora's box means in modern times, it makes a situation whereby it looks beneficial at first, but at the long run, it seems harmful. Firstly, we were taught in microbial biotechnologies first semester that there are some ethical questions, social questions that have been asked about this microbial engineering, about these microbes that have been engineered in the lab, such as who owns these genetically modified organisms? The researchers, what is the risks of them being protected, and are they safe for consumption? Are the environment also saved by these microbes that are genetically engineered? Now, this leads me to my first point, which says biosafety and bioterrorism. Biosafety, we were taught in Micro 2H2 that biosafety is the employment of the protection of the lab, the protection of the lab personnel and the environment from the infectious living organisms. But we cannot say of sure that 100% that these organisms won't slip out or escape. And if they do, we are not sure of how these microorganisms will behave. Also, it is not a new news that microorganisms can be synthesized, as in 2002, Edward Wimmer's group of Sunnybrook University synthesized poliovirus from chemical codes. And so now, what if this is done by nefarious individuals that want to do this for their own selfish agenda? It will breach biosecurity, and also it will lead to bioterrorism. Leading to bioterrorism, it can use and can affect the environment. It can affect life and ecosystem, meaning affecting SDG 14 and affecting SDG 15, which is lives in water and lives on land. Secondly, environmental hazard. When these microorganisms get into the environment, they can release toxins, and these toxins will compete with the beneficial microorganisms in the soil, whereby releasing toxins, and these toxins will depopulate the beneficial microorganisms that are in the soil, whereby affecting our ecosystem, because there are these microorganisms that are important for our ecosystem.
One minute more.
Also, Secondly, they can be resistant. If these microorganisms get in contact with other microorganisms, they can be resistant and turn to superbug, meaning we were taught in Micro 381 about gene transfer, conjugation, transformation, and transduction, whereby a bacterial cell can get lysed and a competent cell can take up that and get transformed, or it can be transduction can occur, whereby the viral particle can attach to the bacterial cell, and when lysis occur, it can take part of that, and if it take part of that, it can act as a carrier to another bacterial cell. If this is being transferred to another bacterial cell, then that bacterial cell is going to be resistant to antibiotics, whereby they are going to be made superbugs. Lastly, health risks. These microorganisms, they are going to be released into the environment, poses an health risk to us, because if they are in the environment and we take them in, if we consume this plant or the product into our body, it can cause an antigenic response. Meaning a foreign matter or a particle is being introduced in our body and it's causing an antigenic response, and peradventure, if you take so much of this, it can lead to cancer and other health implications, whereby these microorganisms, this cancerous cell, I'll use cancer for example, these cancerous cells spread to the bone marrow, and the bone marrow, we're taught in immunology, is a site whereby blood cells are being produced. Now, if they affect these blood cells, it's going to weaken the immune system. In conclusion, it is important for microengineering because it has addressed some global challenges, but we should not ignore the risks, we should not ignore the cons of these, and so we should put in a cautious and well-regulated approach necessary so that the benefits of this synthetic biology do not come at the expense of potentially irreversible harm. Thank you.
Thank you very much. Thank you very much. Without wasting much time, the second speaker from the supporting side will come and present its arguments for 3 minutes, please. 3 minutes. We are trying to work with time. Thank you.
Ladies and gentlemen and honorable invited guests, good morning to you all. My name is Abdullahi Halilu from the Department of Microbiology, Ahmadu Bello University, Zaria. I stand before you today to build on a foundation laid by my fellow debater who spoke before me, and delve deeper into the transformative realm of microbial engineering. As we navigate the complexities of 21st century, it has become evident that traditional approaches to sustainability, healthcare, and demonstrate on this journey of discovery and explore the transformative power of microbial engineering together. While it is true that unintended consequences can occur, microbial engineering can also be built microbial engineering can also be designed with built-in safeguards and containment strategies to minimize risks. Additionally, the potential impact of microbial guidelines and regulations, specifically for synthetic biology and microbial engineering. In conclusion, while the concerns on microbial engineering potential risks are valid, we must not let fear stop us from harnessing its potential. Not let fear stop us from harnessing its transformative power. By acknowledging and addressing these concerns through robust regulation, transparency, and public engagement, we can ensure that microbial engineering benefits humanity without compromising our values and safety. Thank you very much for listening.
Thank you very much for that very brief presentation. Thank you so much. Now, the second speaker on the opposing side, please come forward for your arguments. As well, you have just 3 minutes, so make it snap. Put your hands together for power, please.
All protocols observed. Ladies and gentlemen, my name is Abigail Chongdun Thomas from the Department of Microbiology. So far in this debate, the proposing team have only been able to highlight the benefits that comes with technology while undermining the threats it poses on our health and environment at large. Even though the second speaker was able to say that they are going to ensure that this goes with careful research and also mitigation, you will believe with me that how this involves altering the genetic makeup of microorganisms will actually require a period of time to ensure that this does not pose a harm or effect in the long run. They are only looking at the current situation and not also and undermining the risk it might pose on our health and environment in the long run. My leading speaker was able to highlight the risk involved with microbial engineering, which you will believe with me are too significant to be ignored. To strengthen our argument, I will be taking you through already existing consequences of microbial engineering. To start with, microbial engineering has contributed to the development of antibiotic-resistant bacteria, which pose a significant threat on global health. This leads to more severe infections, leading to longer stay in hospitals, higher medical bills, and high mortality rates, countering SDG goal SDG goal number 3, good health and well-being. Furthermore, ladies and gentlemen, microbial engineering has made it possible to modify pathogens, making them more deadly, more resistant to treatment, and more easily transmissible. This alone poses a serious threat on global security. Imagine a case of misuse, accident, or selfish ambition. Ladies and gentlemen, this counters SDG goal on global security. Furthermore, ladies and gentlemen, the introduction of genetically modified organisms, genetically modified crops, has displaced the traditional agricultural practices. In India, for instance, Bacillus thuringiensis cotton has displaced traditional cotton varieties, although this cotton actually increased yields and reduced pesticide use to death. In fact, in some situations, it has increased the rate of farmers' suicide due to the inequality and countering SDG goal number 10, which says reducing inequality. Ladies and gentlemen, by acknowledging the potential of unforeseen consequences, we can take a more cautious and informed approach to microbial engineering and synthetic biology, ensuring that we prioritize safety, responsibility, and care considerations alongside innovation and progress. Thank you.
Thank you very much. Thank you very much for the presentation. Now you are happy. Now you're happy. All right. Now let's have the first speaker on the supporting side to come wrap up his presentation for just 2 minutes. 2 minutes. The first speaker for the supporting side to wrap up his presentation for 2 minutes. We'll skip it if you're not ready. Time is not on.
Innovation can mitigate these risks. Many of these risks that have been called can be mitigated or diminished to a manageable level through adopting regulations and strict frameworks allow us to mitigate this. As with every great scientific innovation, retroviral drugs, neurosurgery, even social media, they come with their pros and cons. But in the case of synthetic biology, pros far outweigh the cons. In conclusion, synthetic biology offers a powerful toolkit for designing a sustainable future. By embracing this technology, we can create a more equitable, prosperous, and environmentally conscious world for generations.
Put your hands together for him. Thank you very much. Thank you very much. The first speaker on the opposing side, your presence is required here. Come for your 2 minutes to wrap up your points. Thank you very much.
Still standing on the existing protocol, my proponents say that the careful research and investigation of risks of this the risks associated with microorganisms, we can mitigate it. But I would like to tell you that we've not given enough time. We need time to test things. If you get something from the market and you want to make sure that it's very effective, you test it with time, and with time of proving, then you can take it out confidently that you have done your inside research 100%. Now, if you listen to what Professor A.B. Suleiman was saying, he enlightened about a software called SPADEX, which is used for genomic sequencing. Now, if it is gotten into a wrong hand, do you know what's going to happen? It's going to affect SDG 14. SDG 15, SDG 3, SDG 6, SDG 10. Ladies and gentlemen, maturity will tell us that in an argument, not necessarily for a win or a losing side, but for us to be able to portray a balanced ideology on any topic. So there is need for constructive criticism. Thank you.
Thank you very much. Thank you very much. We have come to the end of the debate, and like we said, it's not a competition, it's an educative session. We discovered that the world we live in is more like a ticking time bomb. That if care is not taken, a whole lot of problems will emanate. Chia Ito said something in 2015, that sooner or later, we are going to have more plastics in the ocean than fishes, which is an indication of environmental pollution. Global warming is on the rise. Antimicrobial resistance is making its way deep through our economy, and as a result of that, we are at the mercies of different aspects of scientific research. So we decided on bringing in this topic, so we will deliberate. We'll see the pros, we'll see the cons, and chart a way forward. Thank you very much for your time. Without further ado now, I will call on my mother, in person of Dr. Habiba Atta, for the citation of the third speaker. Thank you very much, and put your hands together for her as she comes forward.
Good afternoon again, everyone. Permit me to stand on the existing protocol. I would like to invite Dr. Fatima Mohammed Musa to the podium. Dr. Fatima Mohammed Musa, please can you join us on the podium? So our third and final speaker is Dr. Fatima Mohammed Musa. She is the current head of department in Kaduna State University, Kaduna. And one of the first things you notice when you come to the Department of Microbiology, you see a lot of babes and women around. So definitely we must have a female speaker, and no other person better than Dr. Fatima Musa to do that. So she has a Doctor of Philosophy degree in Medical Microbiology from Ahmadu Bello University in 2017. She has a Master of Science in Medical Microbiology in 2002 and a Bachelor of Science degree in Applied Biology in 1988, both from Bayero University, Kano. Dr. Musa has had over 20 years of experience and practice in teaching biology, microbiology, and also in conducting other French research work practices in the Department of Applied Sciences, College of Science and Technology in Kaduna Polytechnic. She has supervised over 100 HND projects and taught IJMB courses in the School of Preliminary and Remedial Studies. A round of applause, please. She rose to the rank of Chief Lecturer in 2012 and later transferred her services to Kaduna State University as a Senior Lecturer in 2019. She has taught medical microbiology courses both at undergraduate and postgraduate levels. She rose to the rank of Associate Professor in 2023. She has supervised over 40 projects at undergraduate level, 20 postgraduate projects, and currently supervising 4 pioneer doctoral students. She is also currently an external examiner to many postgraduate students in Nigerian Defense Academy and also in Ahmadu Bello University. She has over 50 peer-reviewed journal publications and has attended over 20 scientific conferences at national and international levels. She has also served as a book reviewer and a reviewer in some learned journals, notably Nigerian Journal of Parasitology and Public Health, which is published by the Parasitology and Public Health Society of Nigeria. Dr. Musa has headed some administrative positions and has served as a head and member of many committees, some of which are the PPSN State Chapter Coordinator between 2012 to 2022, and also as the head of the Department of Microbiology in Kaduna State University from 2020 to date. She has coordinated many scientific and collaborative researches that has led to the establishment of the Center for Malaria and Mosquito-Borne Diseases Research and other related researches in Kaduna State University. She has also coordinated workshops, conferences, and symposiums, both within and outside the department in Kaduna State University. She has facilitated the hosting of the recent Nigerian Society for Microbiology Northwest Symposium by Kaduna State University in 2023, and most recently, the forthcoming national conference scheduled to hold in September 2024 at Kaduna State University. Other experiences in her career revolve around the design, implementation, monitoring, and evaluation of health programs and projects, with the ability to deliver assigned responsibilities. She has received several trainings as a facilitator and supervisor with support from the State Ministry of Health, and also in collaboration with Partnership for Transforming Health System 2 from 2008 to 2010. Other experiences include policy influencing social issues, situation and technical health researches, a Millennium Development Goal Project 2012, as well as knowledge on the epidemiological characteristics of COVID-19 epidemic Kaduna State. All this was through the Kaduna State COVID-19 seroprevalence study in 2022. Her area of specialization is medical and pharmaceutical microbiology, with specific research interest in medicinal plants and antimicrobial properties. She is happily married with many children, and she also has grandchildren, which happen to be twins. So please help me to welcome Dr. Musa.
Thank you very much. I'm highly honored for this opportunity to come and present a seminar, especially in this prestigious university that actually is one of the best universities in Africa. And I'm always proud of this university. Well, actually, because of time, this is going to be one of the shortest seminar I'm going to present. So, based on the topic given to me, I'm supposed to go directly to talk about how we prepare our future scientists, future microbiologists for emerging trends and technologies. But I'll just state the important points. So, all protocols duly observed. The topic for presentation for today is preparing future microbiologists for emerging trends and technologies. So the field of microbiology is rapidly growing with the advent of technology, as well as social needs that warranted the need, as well as the emerging global challenges. Then emerging trends in microbiology are, however, new developments or techniques that have been used in researches, and also the application of these technologies that will indirectly impact on our health and industries. So such emerging technologies also has to do with the innovative tools or techniques that are currently being developed, and which is always made available within the foreseeable future. So current molecular methods use genetic testing, epidemiological studies, typing, as well as in-situ hybridization, culture phenotypic and biochemical tests due to complexity and variability of specimens. Such specimens types are tested order dictate for the fact that because they are very cumbersome, and for instance, you want to culture fungal or bacterial culture, you need to undergo so many series of cultures. So based on that, we also have some of the characteristics of emerging technologies. These characteristics are usually novelty, that's the early stages of the development, meaning that such technologies are usually new, usually originates from a specific place or a person. Then such characteristics of technologies also provide impact, interdisciplinary nature, it is rapid in advancing, and examples like artificial intelligence, biotechnology, nanotechnology, and renewable energy technologies are some of the emerging technologies. So now, the major focus of today's presentation, as I earlier said, is very simple. We need to prepare future microbiologists for emerging trends and technologies. So this research, for instance, this future microbiologist needs to be nurtured from the grassroots. for it, we need to co-opt the interest before a microbiologist can even be given many opportunities to explore. Such interest has to do with the basic level. That's showing that's a co-opting the basic knowledge of microbiology, and then after that, enable students, give students the opportunity to explore by themselves about the real impact of what microbiology all entails. For instance, research opportunities should be given. This will provide access to research projects that will explore cutting-edge topics and to allow students to engage with real-world problems and innovations. There should also be workshops and seminars of this nature. By organizing workshops, it will actually give students opportunities, the opportunities to explore their capabilities as well as their knowledge, and then enhance their technical knowledge towards the subject. There should also be research. There should also be organized events featuring experts in emerging technologies. Such events will also provide students with advancements and career opportunities. There should also be soft skills development. Such soft skill developments will allow students to communicate as well as enable them to have essential collaborations with innovators. Such collaborations should be experts from various fields, either in the field of engineering, computer, as well as other areas in sciences. We also have internship and industry partnership. Such internship partnership will be a collaboration with the companies as well as research institutes or governmental agencies so that they can give practical experience in applying their knowledge. There should be continuous learning through certification on online courses as well as conferences to keep pace with emerging trends. In conclusion, variety of techniques are used to study microorganisms, and these microbiologists also develop and apply new technologies, leading to rapid advancement in the field of microbiology. Including these technologies in microbiology programs or curriculum or laboratory settings and other activities like mentorship, research trainings, workshops, seminars, and conferences is essential to prepare future microbiologists for the rapid evolving field of microbiology. And by combining the strategies or features mentioned, microbiologists will be well equipped to navigate and contribute to the evolving landscape of their fields. So thank you for listening.
Thank you very much, Ma. Thank you very much, Ma. We appreciate your presence and thank you for the wonderful speech. Now, before the question and answer session, I looked onto my left-hand side and I discovered that one of our seasoned lecturers is also here at present. And ladies and gentlemen, I would like us to put our hands together for Professor Doku. Let us stand up for recognition, sir. Stand up for recognition, sir. Thank you very much, sir. You're welcome, sir. Thank you so much. So now, for the questions. Yes, our chief technologist as well, in person of Dr. Eno Adai. Thank you very much, ma'am. All right, we have some questions here, and we have a whole lot of questions actually, but due to time constraints, we reduce them. So, for the very first speaker, sir, as a 400 level microbiology student, can you please give a concise blueprint for a student interested in venturing into bioinformatics with the PSc in microbiology?
Okay, thank you very much. You know, what surprised me is that there are universities that are running bioinformatics at undergraduate level as courses. I taught India for almost 5 years. They're running bioinformatics at undergraduate level. That is one. Secondly, most of the biological department outside Nigeria, mostly South Africa, all of them are running degrees in bioinformatics. That is it. And then I think presently, we want to start doing something here that we may have a bias degree. One of the departments here in ABU will be awarding bioinformatics as a degree. Now, bioinformatics is simply one of the tools you use for analysis. That is a fact. So it can come in under biotechnology as a degree, but normally it can stand on its own because it's all about computational biology. You are doing computation on macromolecules, so it's almost everywhere. Once you have a degree in microbiology, some biological sciences, you are qualified to run MSc in biotechnology. I hope I answer your question.
Thank you. All right. The other question here, sir, says, how do researchers use phylogenetic analysis to track the spread of infectious diseases and the limitations? Yes.
Okay. Number one, we are talking about phylogenetic analysis. I gave an example of certain software. For example, we have one algorithm that uses the high climbing technology. What it does is simply reconstruct the sequence or the DNA of a genome and look at mutations within it and do DNA visualization with other organisms that may be related from evolutionary studies. Then they can now compare and do prediction on how long can that new organism survive as a reference to the other organisms that are available. They look at the homology, how similar are they, or how different they are, and then they infer, just like what we're doing, we want to detect functional
intelligence epidemiology.
Yeah. Number one, all activities. I mean, one of the persons who participated
in the debate
was talking about on the quality or mistakes. None of any of the software, algorithm, mathematical modeling, statistical modeling that is released for people to use without quality assurance and quality confirmation. They must make sure it has passed a level where they can say it's excellent. And they must show that to the best of their ability, that thing cannot affect the environment or the people that are using it. So it's not just about a hypothesis. It's something above proving it, accepting it as a law. It takes a long time before a certain software or a certain law will be passed that every other person can practice it. No, it's more than that. And let me tell you, one of the expert thing on IA is everybody is using it. Meaning, if today you are online, you are using it, other people they see and they know what you are doing. That is why we talk of collaboration, so that we can cover up disease or can bring something new. So once any other lab, look, we have 3 very important genomic labs in the world. We have the European Molecular Biology, right? Assembly. We have the GenBank, we have the DNA of Japan. EDDNA, that is Japan DNA repository. All these people, they work almost the same. They work on primary databases, they work on secondary databases, they work on specialized databases. And most of the things they share is common. So once they realize something is different from what has been deposited, it's simply a question, and somebody will pick up. Don't forget, not quite long, some scientists in China who has been publishing flowering publication in one of the high impact journals. He came up himself to say, look, what I've been writing actually is false. You understand himself? Yeah. So all his journals were removed and his titles were removed. You understand that he was like man just alone. So these things are not just done in a single form. You cannot just be doing it alone in such a way whatever you do you feel is okay. No. A lot of people are examining what to do. This thing is an evidence-based technology. Do it, they say. Everybody asks questions. Okay? So there is no mistake. There are a lot of things to do, a lot of activities that is for public to use.
Another lecturer in person of Dr. Emesali for some questions. He was actually not one of the speakers, but there are some questions that we believe he is Dr. Emesali.
What has to be done to reduce the impact of infectious diseases and predators on farmers' production system? Well, I think if you have listened to the presentation by Dr. Bishir, he must have answered this question. He talked on pest control by the use of bacteria, specifically Bacillus thuringiensis. It can be used to control pests as well as to genetically produce resistant varieties of the seeds that are resistant to the pests and other diseases of plant. The second question is why some cancer, like cervical cancer and smokers' lung cancers, aren't classified as infectious diseases. Well, from your epidemiology, you are told that an infectious disease is a disease that is caused by a microorganism, either bacteria, fungi, virus, or whatever. Now, if the cancer, as you have said, smokers' lung cancer, is not caused by a microorganism, or the cervical cancer is not caused by a microorganism, it cannot be referred to as an infectious disease. Because infectious disease, as we have said, are diseases caused by microorganisms. But is what are the future possible occupational roles for microbiology students? I think this too has been answered by the last agriculture and the health, wherever you can fit as a microbiologist. And then the last question is, why is medical laboratory science, MLS, referred to as the higher version of microbiology and broader spectrum of chances than the medical laboratory science? Thank you. These are the only questions I have.
Thank you very much. Thank you very much. All right, Professor Abi Suleiman has something to say in regards to that.
Why most of you are so worried? No. The medical laboratory science is normally used to be, the name is medical laboratory technologies, meaning it's a technician kind of a job. One. Secondly, their training is based on middleman powership anywhere. That is all about diagnosis, which is less than a course unit under medical microbiology. The reason why most of us are worried is because there is no good scientific job where microbiology can fit. How many areas do you have? Environmental, industrial, agriculture, medical, petroleum. Understand that? All these are areas where you can go and dance and talk comfortably. But you are so restricting yourself to go to the lab and somebody is your boss who will give you a sample, you check and write your report and send, and that's the end of the story. Because there is a job, we should pray to be more developed in this area. And for your information, I was one time the deputy secretary of the Nigerian Society of Microbiology, and I have been one of those people who are fighting for what? Professionalization. That is when they always do professionalization. Meaning you are soon, because you've done the second reading on that, you are soon going to be licensed to practice whatever you want in the different areas of specialization. Thank you very much.
Thank you very much, sir. Thank you very much, sir. Quickly, we are trying to wrap this up before 1 p.m. The second speaker, we also have some questions for the second speaker. I'll briefly read them while he does justice to them in a matter of a few minutes. Now, the first one goes, how does Pseudomonas contribute to bioremediation process? And what specific metabolic capabilities enable it to degrade toxic compound?
Okay. Thank you for the question. Someone is asking about how Pseudomonas is able to contribute in bioremediation, especially in toxic environment. There are some reasons, I think about 3 I can mention. One, that because of its adaptability, So it is able to survive in so many environments regardless of the environmental composition in terms of toxins, heavy metals, and some chemicals. And secondly, because of the varying metabolic capabilities or metabolic networks. So Pseudomonas has so many metabolic pathways that if this is blocked, then it can look for another pathway in order to utilize the materials present in the environment. So because of that, it is able to break down so many compounds in the environment, and that's why it is used in bioremediation. And also, by the virtue of some compounds which we call biosurfactants, they are more like detergents, which are produced by Pseudomonas, especially Pseudomonas putida and Pseudomonas aeruginosa. For these biosurfactants, which are more like detergents, when they are produced by Pseudomonas, it tends to break down some hydrocarbons into usable products which can be further broken down by other organisms, even if Pseudomonas cannot break them down. So these are some of three characteristics of Pseudomonas. That's why it is used in bioremediation, especially in water, environment, and soil.
Thank you very much. The second question says, what are the recent advancements in synthetic biology that are driving microbial biotechnology?
Okay, I think towards the end of my presentation, I mentioned that there are some future trends in synthetic biology that are applicable in microbial biotechnology. I think I mentioned microbial biosensing, which is usually used in detection of pollutants in the environment. And also there is what we call bioprinting and biocomputing, which is used in synthesis of tissues and organs using microorganisms. And this can find application in regenerative medicine, especially if someone has heart failure or any tissue or organ failure, then microorganisms stand chances of being used to regenerate these organs for application. And also, I mentioned CRISPR-Cas technology, which is basically used in gene editing. So all these are synthetic biology tools that have applications in microbial biotechnology.
All right, thank you very much. Put your hands together for him as he takes his seat. So, quickly, the next item on the agenda is a short speech by the President of the NAMS in person of Comrade Abdulrazak Shaibu. Put your hands together, please. Put your hands together, put your hands together. Abdulrazak Shaibu is a friend with whom we go way back. First met him in 2017 when we were still students at the School of Basic and Remedial Studies, SBRS. And over time, from that period till now, he's been a role model and mentor to a whole lot of students, ranging from medicine, pharmacy, microbiology, even down to the physical sciences. He is a young man with a very big influence. So ladies and gentlemen, NAMS President Abdulrazak Shaibu.
Respected guest of honor, our Madam, the Dean, I speak of her mission and vision. I speak of her honor and valor, an icon of hope, a beacon of integrity, a symbol of easygoing, an attentive person, a bold, brilliant person, a charming person, a diligent person, an expensive person, of course, an extremely extraordinary person, of course. I speak of women that gives the younger generation a hope and even a chance to dream high. I speak of our mother in person of Prof. Mariam Aminu. May I use this opportunity as well to recognize the head of department of microbiology, our father, of course, Professor M.B. Tijani. May I use this opportunity as well to recognize our mentor, our tutor, a role model, of course, a true definition of a leader, in person of the young ambassador, American Society for Microbiology to Nigeria, Mr. Yusuf Omodapajiti. Without wasting much time, ladies and gentlemen, all protocols are duly observed. I want to use this opportunity to thank the convener of this very important conference. I believe the students, all of us here, I believe we are moving with lots of knowledge, with lots of ideas, with lots of education, with lots of values from this important conference. I must also thank the moderator. She has been there for us. Her tireless effort is something that we can never forget. Her support, her dedication, her determination, and devotion are beyond imagination of a lad. I must, of course, give credit to our mother in person of Dr. Habiba I. Atta. Ladies and gentlemen, I don't want to go deep. We have limited time and today is Friday. I just want to use this moment to thank each and every one of you for coming. Most importantly, the best students in ABU, in Ahmadu Bello University, and the student are the student of my Department of Microbiology. Yes, I have many more to defend the students. If you talk of brilliancy, if you talk of humility, if you talk of respect, if you talk of responsibility, if you talk of dedication, if you talk of determination, if you talk of devotion, if you talk of hard working, if you talk of sacrifice, of course, you have to mention a department, and there is no any other department that should come first beyond the Department of Microbiology. So, ladies and gentlemen, I want us to please use the knowledge. Our guest speakers here, our mother coming all the way from Kaduna. We have the NAMS president all the way from Kaduna State University seated here. We have the governor, NAMS Northern Region, representing over more than 19 institutions. We have the NAMS president from Federal University, Dutse. They all came here to learn. So I believe if someone can come from very far place to attend this conference, I see no reason by which us, that it comes to our home, we are going to miss this. So please, let's use this knowledge to build our future, our academic journey. Respected ladies and gentlemen, I must also commend our lecturers from the Department of Microbiology. When you talk of easygoing, when you talk of cordiality, when you talk of hardworking, when you talk of knowledge, of course, you can't go and touch our lecturers. They are fully loaded. Yes, they have been giving us our best from the very first day. I must also use this opportunity to please appeal to our lecturers more again, to be merciful with the students and their struggle. Students have been going through a lot. We believe you have been doing your best, but I will also appeal at the student president. The lecturers should be more merciful with the student, more generous with the student, more approachable. Yes, you have to be approachable to the students so that they can meet you at any point to give you their complaints, to their concerns and grievances. As well, I will also use this opportunity to remind our lecturers their kind words are very, very important in our life. So we please urge you to continue giving us those kind words, kind correction. Even a smile face is a very good thing that we should have as well. So I believe our lecturers here are hearing me, and I believe they are going to be more approachable, they are going to be more patient, they are going to be more considerate with the students. The students are going to do a lot. I can speak to you. We have many students here, but I can bet there are people that came to this venue without taking breakfast. Students are going through a lot. Even the Gari that have been termed as a student power, it was common before, but it's very scarce now. Even the airlines are finding it very difficult to obtain Gari. So, ladies and gentlemen, though the MC is reminding me that I have 2 more minutes, I would like to just outline a few of my achievements in less than 6 months. In less than 6 months in office, I have achieved a lot. More than 30 projects within 6 months, of which 40% of the deals were used, while 60% of the funds were raised by my friends, relatives, and others. So we have fixed the spoiled desks in the classrooms. We replaced the stolen curtains in 400 level with new ones. We repaired the spoiled windows in 400 level. We fixed all the wall clocks across the classrooms. We supplied water for the first time from the overhead tanks to male and female students' toilet on the ground floor and the male staff toilet on the first floor through a new connection, which has not been operational for over 6 years. We repaired the faulty fans across classes in collaboration with our mother. We replaced the faulty sockets across the classes. We replaced the faulty bulbs across the classes. We repaired and provide locks for the 6 doors in the male and female student toilet. We purchased Dettol, kettle, and buckets for the toilet. We fixed the light issue in the female toilet that have been non-functional for about 6 years. We established the NAMS Public Health Awareness Team. We supported the NAMS academic team with tools like hard drives and others. We provided constant tutorials for all the classes with special welfare to tutors, giving them data and some stockings and students during the tutorials. We conducted orientation for 100 level and DE students. We organized a virtual service orientation through Google Meet and physical orientation on essay questions approach for 300 level students. We provided data and airtime allowances for the first time to all the class representatives across from all the classes in my department in the first semester. We sent data and airtime to them. For the first time, we took NAMS to Twitter, official Twitter account, IG account, and Facebook. We repaired the NAMS complaint box and created a complaint email for students. We supported the casual workers with more prompts and motivation tokens at intervals. We fixed the logs in 200 and 300 level classrooms. We conducted service defense mock presentation and project guidelines programmed by the academic team. When Ramadan came, I called my friend studying medicine in Egypt. We used to do one support during Ramadan. He sent some tokens, and with contribution from my own self, we bought 1 bag of sugar and 10 care of debino and shared to the student in microbiology. We facilitated the purchase of 3 fans for the classes by the then HOD, Prof. Mariam Aminu, our mother, who is now the Dean, Faculty of Life Sciences. In extension to this, he supported us with extension wire for the library and bulbs around the department. We renovated the male and female toilet in collaboration with Niles Zaria. We supported the weak and sick students in most need at intervals, buying them some tokens and also calling their parents to check on their well-being. As I'm speaking to you now, we have created NAMS e-library where we have updated all the materials from 400 level to from 100 to 400 levels, which will be much needed. Respected ladies and gentlemen, these are just few achievements in less than 6 weeks. The main purpose of I outlining this is a challenge to those coming. Leadership is an opportunity. Almighty give you the chance, you should try and change the life of people in you. Try and use the opportunity to impact. Being you a teacher, being you a casual worker, being you a classroom, try as much as you can and put smile on the faces of those you are leading. Ladies and gentlemen, thank you for the audience and thank you for listening.
Thank you very much, Mr. President. When it was coming up, I said it's a young man with a very big impact, and you guys have seen it. That's just the little of what we've experienced, and believe me, more are still undergoing. So, ladies and gentlemen, allow me to introduce the House Association of Bankasa, right? I'm Ibira. We'll say Adai Sakasaka, our overall father. So, ladies and gentlemen, allow me to introduce the head, Department of Microbiology, in person of Professor M.D. Tijani for the closing remarks. Thank you very much, sir.
Vice Chancellor, ably represented by the DVC academic, Professor Raymond Bako, the Dean, Faculty of Life Sciences, and the entire team in the deanery, the convener, Mr. Yusuf Tajuddin, the representative of the ABU alumni, in person of Malam Bashir, the Deputy Director, Directorate of University Advancement, the organizers, meaning the NAMs and their team, the students, invited guests, the entire team of the Public Affairs Division, the security officers from the ABU Security Division. Good afternoon. It is actually an honor and privilege for me to give a vote of thanks for the closing remark of this August occasion. I sincerely thank the convener for choosing Ahmadu Bello University Zaria for this particular occasion. And honestly, in the course of the occasion, I have to sincerely extend our appreciation to our Vice Chancellor, the Vice Chancellor, because despite the fact that he has a very tight schedule, he's unavoidably absent here, he was following us online, that he even communicated with us while the program is going on, and he has to send his representative to come and represent him. We thank you very much, the Vice Chancellor. I also want to sincerely appreciate the Nigerian Society of Microbiology, or the Nigerian Association of Microbiology Students. Honestly, I feel highly honored for this wonderful feat they have achieved. And I want to sincerely also thank every invited guest that honored our invitation here. We also look forward to seeing you in the forthcoming conferences. And for the 400 level students working on their research projects, have it at the back of your mind. There are 3 conferences coming up, all of which you can present or showcase your findings. The first one will be coming at the end of this month, the SAM conference in Lokoja, starting from the 25th of August 2024. The NSL is also coming up, and luckily enough, we have the chairman of the organizing committee in person of Dr. Fatima Musa, who is the head of department. That conference is also coming up in September, starting from 3rd September. And for those of us, the virologists, the African Virology Conference is also coming up in November and is starting from 25th of November 2024. So we are looking forward to see all of you there showcasing what your findings are. So I wish every one of us that honored this invitation safe journey back to your destinations. Have a great weekend, and thank you once again.
Thank you very much, sir. Thank you very much, sir. The media and publicity section of the institution just gave me wonderful news right now. They said the whole session was streamed live. Go to YouTube, go to Twitter, Instagram, Facebook. We have this whole program streamed live there. So I'm urging us now to quickly on our mobile phones, if you know, wait, let me even ask, did you enjoy the program? Did you learn something new? I learned if there is a word like synthesis, because the things I learned that I actually knew that I didn't know of before now were in multiples. So quickly on YouTube, you can check Ahmadu Bello University Zaria. The video is there, you like and you subscribe to the channel, okay? Or you can check at Ahmadu Bello University. The single word, no spacing, at Ahmadu Bello University on YouTube. The video is there for you, so you will enjoy it. And thank you very much once again, distinguished lecturers, invited guests and speakers all the way from Kaduna. Thank you, the American Society of Microbiology representatives and the young ambassadors to Nigeria. Thank you. So the last item on the list of events is now photographs. So from the media and publicity section now, we will take photographs with the members of the high table. Then I think it will be okay if the students progress in an orderly fashion and take probably one shot with members of the high table as they take their leave. All right, members of the high table will have to come over here for the picture, the photograph. That will be the last thing. All right, let's quickly do that. It shouldn't take much time. Let's quickly do that. Then, ladies and gentlemen, please, you should be reminded, for those of us who attended and registered for the conference, there is a certificate of attendance. So please, check your email, or at the same time, you can meet with your individual class reps, okay? They will forward it for you. Just meet with them. There is a token for it, actually, and it's of utmost importance, okay? That will be one of the proof you are going to have that you were a part of this wonderful event. When they are calling names of people who made history, names of people who were there when NAMS and ASM collaborated for the very first time in the history of mankind, your name will also be mentioned and you'll have a proof of that. So please reach out to your class reps, reach out to myself, reach out to any NAMS executive you know of, and we will direct you on what to do in terms of getting the certificate. And at the same time, there is, for the Muslim brothers and sisters, we have provided water for ablution. So we'll perform ablution before. So please take another one. I'm still right here. Don't forget about the channel. Don't forget about the YouTube channel, the Ahmadu Bello University Zaria channel. Okay. The students in an orderly fashion. I don't know how you see it, but from where I'm standing, I can say that NAMS ABU Zaria, we have made history. I strongly believe what we did today will forever echo in the halls of history of mankind, as bringing ASM all the way from the United States of America down to Nigeria, ABU precisely, and sharing important knowledge and insights into the future perspectives of microbiology students. Thank you very much for your attendance. Also, for the Muslim brothers and sisters, please be reminded, we have provided water for